Thank you. We'll move on to the next speaker, Dr. Cassidy Rist, on ivermectin roadmap, enhancing impact from One Health strategies. Come on up. You have to just push really hard. Push really hard. Yeah, that one.
Okay, thank you. Okay, did that work? Yeah, great. So what I'm going to discuss today is how we might enhance the impact of ivermectin as a tool for malaria elimination through its use in livestock populations. And some of you are probably familiar with this, have heard of it. And I know that probably everyone in here is really familiar with the Mass Drug Administration in humans, and has heard about it in livestock, but I'm hoping to kind of bring it to life and talk about it from the perspective of some of the more intricate details that we might need to consider as far as risks and benefits to using this as a tool. So this is a lovely picture that I'm sure you've all seen of residual transmission and the different points that we might intervene to reduce that transmission. What I'm, of course, interested in is this little guy here, the cow that's depicted, and what he represents. So what he really represents is a huge source of protein for zoophilic vectors of malaria. So the average cow, adult cow, is about 350 to 400 kilograms in weight. That's a lot of blood, and that's a lot of surface area that mosquitoes can feed on. And although the livestock that we're considering don't become infected with malaria, as I said, they serve as a nice blood meal that can help keep the population of mosquitoes that transmit malaria alive. You're right, it is hard. So the key points that I want to talk about from a veterinary perspective is that ivermectin has a long history of use in veterinary populations. So we veterinarians have been using it for over 30 years in cows, sheep, goats, pigs, horses, and it's a really important tool for us to control intestinal parasites, lungworms, as well as ectoparasites in animal populations. And this is really essential because improving animal health maintaining animal health is important to populations who rely on them for food security and economic security. What we know is that the PK properties are well characterized in animal populations. So as far as it being effective for the indications, veterinary indications, they're extremely effective as well as quite safe. So doses of up to 30 times the labeled dose for subcutaneous injections have been proved safe in cattle, and similar studies have been done in ruminant species as well as swine. So it's effective and safe for veterinary indications. So I showed the one image that Carlos said he has continued to show, but I wanted to do this to just reiterate that when we're talking about treating livestock with ivermectin, that we're really doing this to treat more sources, to cover the number of blood sources more generally with ivermectin. So every warm-blooded animal, including humans, that a mosquito could bite could be sort of poisonous to these mosquitoes if you have levels of ivermectin high enough. So efficacy in livestock, so this is not, I was speaking about the fact that we know it's efficacious for veterinary interventions, but this slide is talking about efficacy as far as its ability to kill mosquitoes. So this is a PK curve for, in the red, you see the red dots, that's for an injectable, a single injection given to pigs at a labeled dose of 200 micrograms per kilogram. The orange dots are a single dose injection for cattle at 3.15% solution at 600 micrograms per kilogram. And what the blue line indicates is the blood concentration that's required to kill greater than 95% of Anopheles arabiensis within 10 days. And so in cattle, we can see this blood level is maintained for up to 6 weeks using this long-acting injectable, and in swine, it's about 1.5 weeks. So we know that we can get blood levels up high enough to have impact on mosquitoes, at least this particular species. So when we consider the likely drivers of efficacy in livestock, so efficacy of using mass drug administration in livestock, certainly we have to consider the local species that are present. So are there species present that actually bite livestock? So are they zoophilic versus or zoophagic versus anthropophagic? And what scale do they range on that? And this is a really nice, you can't see it perfectly, but basically I'll describe it as this is the proportion of blood meals obtained from humans. The left is 0% up to 100%. And this depicts the various species that can transmit malaria. And we see that some of them, specifically those species in Africa, the Arabiensis, has a wide variety. So depending on where you're selecting these mosquitoes, potentially up to 100% could be feeding on livestock, whereas a large variety of them, it's between 20 to 80%. Right? So again, if we want to use this in livestock, we also have to say, are the mosquitoes that are present that are transmitting malaria biting these livestock that we're interested in? Other drivers, of course, include the livestock species and what kind of groups we would select. So when I say livestock, we often think of cattle. That's probably where the most studies have been done. But we need to consider small ruminants and swine as well, depending on the populations and the type of animal species that are most utilized for food and income in those populations. The proximity to communities, so depending on husbandry practices, some livestock are free roaming, whereas others are kept close to the house and are close to human populations. As well as we know there are people who travel, or nomadic populations who actually move from location to location with their livestock. And then, of course, the issue of livestock to human ratio. So this idea of density. Is there a density that would be most appropriate in order for MDA in livestock to be effective? So as far as research and development questions go, as part of the roadmap, really trying to think through this and say, what are the main livestock species and populations to be targeted in the areas that are under consideration? And this really also includes issues of coverage levels, so just like in humans, so very similar concepts apply. Is there a threshold of coverage that we need within a population in order to have the effects that we want on human outcomes? And then is there, we have labeled doses that we know reach or generate blood levels that can kill mosquitoes, but is there a dose and a regimen, similar to all the issues that Carlos just brought up with the human dose and regimen, in livestock that would be most appropriate? There's also this idea that is there a way that we could create a tool, an effective tool that could map veterinary and husbandry practices in a way that could rapidly identify herds that might be at highest priority for MDA? So some of the potential challenges associated with this, conceptually, we don't know that this is true, but conceptually there's the possibility that if you had two vector species in an area, one that primarily bites humans, and they were lower in numbers, and one that was able to bite humans and livestock, if you just treated livestock alone, you could actually drive up or create a niche for the population that only wanted to, or that was only biting on humans, and in effect actually drive up transmission of malaria in human populations. So again, this is conceptual, but it is a possibility. However, we could certainly mitigate that by just saying, hey, if this is something that we're concerned about, an option to prevent that from happening would be to just consistently use MDA in livestock in conjunction with that in human populations. Another topic that I think a lot of people who are not veterinarians don't consider is this issue of food safety. So currently the use of ivermectin, which again is used broadly in veterinary medicine, requires meat and milk withdrawal times. So as a veterinarian, if I give a dose of ivermectin to treat intestinal parasites in a cow, there is a specific waiting period that must be maintained before that cow can be slaughtered for human consumption, or before the milk from that cow can be used for human consumption as well. And this is standard. This is something that is, there's sort of global standards around this. higher doses mean longer withdrawal times, right? So if we were to say that a higher dose was more effective for our outcome of interest in reducing malaria, we would have to consider that that might have some economic impact on the fact that then people could not slaughter their cow or use the milk from their cow for potentially extended periods of time. So these are things we need to consider as we move forward and think about the dose and regimen that might be most appropriate. One thing I did want to bring up, which often is brought up, is there are alternatives to ivermectin. So this is aprinomectin. This is used currently in dairy cattle because there's no withdrawal time. So I can give a dose today, and tomorrow you can use the milk from that cow. So economically, for livestock producers, that might be a more enticing use. However, it's important to consider the cost differences between these two drugs, which you can imagine is significant. So that is sort of this idea of what would we need to know if we think this works, if we think MDA in livestock could work, what are the things we would need to consider, right? So mosquitoes, where they're biting, how does that overlap with livestock populations, the husbandry practices associated with it. So there's some very technical things as far as implementing MDA that we would need to consider. But I think it's also important to think about the indirect benefits and risks outside of those that I've already talked about. And there's a lot of them, I should say, that as Gina mentioned, there's a lot of things that we could talk about, but I want to talk about 3 specifically. The first is economic impact. The second is the potential for development of resistance. And when I say resistance, I'm talking about resistance to resistance in the parasites that I am most concerned about as a veterinarian. So what these drugs are used for in livestock populations. I know resistance to mosquitoes is an issue. There's resistance of the parasite itself, the malaria parasite itself. I'll leave that to other experts in the room who can speak to that more directly, but resistance is an issue of concern for us as veterinarians who are worried about maintaining this drug for use in animal health. The third thing that I'll discuss is this idea of environmental impact and how use of this in a mass drug administration way might impact the environment. So when we think about economic impact, I have here depicted a sort of just a description of the economic production cycle, right? So we have human capital, physical capital, natural resources that are combined in the production cycle to produce income, food, and other goods. And those things that are produced are reinvested to help build capital, and anything that's excess above what is needed to maintain that cycle results in accumulated wealth, right? This is sort of the economic vision of how wealth is generated. And we know that human health is an essential component to this, right? Humans are living beings, their growth, their biological processes are essential, especially contributing to the human capital component of this production cycle. And so as we have healthy humans, they can contribute to this cycle. We get wealth generation that also leads to enhanced human health. So when you introduce infectious disease into this scenario, I think we all recognize that it impacts economic productivity, right? It impacts it as we lose our health, we can't go to work, we're not physically able to do the things that we need to do. Children are not growing appropriately, they're not able to achieve education levels in the way that we expect. And so it's this very clear thing that everyone in this room understands. If you don't have your health, it's very hard to maintain your economic security. So I would argue as a veterinarian that livestock have a very similar role, right? They are living beings that contribute to this productivity cycle, at least in those communities and in those populations that rely on them. And so through this contribution and the fact that they are living organisms that can succumb to infectious disease, when they become infected, they basically have the same effects that human disease can have on this economic productivity cycle. So diseases that afflict livestock threaten economic productivity in households in ways that are really comparable to human disease. So the key points about this is that ivermectin is, with that concept in mind, ivermectin is an essential tool for maintaining animal health, and we know that maintaining animal health is really important for generating these indirect benefits on human health and economic security. And it's important to include these sort of indirect benefits in any sort of cost analysis because it provides a cross-sectoral approach, right? And what this can do is really give us the potential to support additional sources of funding. So we need the agricultural sector on board if we're going to be using this in mass drug administration. We need the agricultural sector on board, and if we can show greater societal benefits to animal health and economic productivity, there's the potential to spread the cost of this across different sectors. The other thing I'd like to bring up is the issue of resistance. And as I mentioned before, not in the mosquito, not in the parasite itself, but in those veterinary parasites that we are concerned about, that these drugs are primarily used for. And so it is a growing issue. There's a lot of documentation that ivermectin and animal helminths are developing resistance. But we also know that it's challenging to identify resistance. So even though we know there's some individual studies out there, the global picture of resistance is really not clear. It's challenging to identify because the techniques that we have are pretty crude. It's basically checking a fecal sample for the number of eggs that are in that sample, giving a dose of the drug you're interested in, and then checking back in 3 weeks to see if you were able to reduce the number of eggs by 95%. So this is very crude. It's phenotypic, which is valuable, but it doesn't allow you to see resistance sort of sneaking up. So this is a concern for us, that potentially we could say, oh, we're not having a problem, but that resistance could appear at some point in time after it's too late, after it's sort of been brewing for a while. I would like to say, though, that there are, I don't want to be doom and gloom about that, there are strategies that exist that because we're seeing resistance in these intestinal parasites that are important to animal health, there are strategies that exist to mitigate this. So similar to human helminth resistance, you can do drug combinations, but something that's unique to sort of the veterinary population is this idea of refugia. And just really briefly, I put up a cartoon up here to kind of depict this idea. This is an example. So in this visual, the little red worms are those that are resistant. The orange ones are those that are susceptible to ivermectin. And in the first scenario, all animals are treated. So every animal in the herd is treated. When all the animals in the herd are treated, the only parasites that remain inside the animal are those that are resistant to the drug. Right? And so then as that adult worm inside the animal that is resistant to the drug produces eggs, the only eggs that are being put out into the environment are those of resistant parasites. And so you see, let me make sure I get the correct, there we go. You can see here that initially you start off with a population of about 8% resistance, and you end up with about 27% of the population of parasites in the field are resistant to ivermectin, right? And this happens actually fairly quickly. In the second scenario, which is the idea of refugia, it means that you leave some percentage of animals untreated. That can be anywhere between 5 and 10% of animals left untreated. In this scenario, we can see that you start off with a population of resistance of 8%, and it ends up only at about 9% through this cycle. Now, you will still get resistance, and resistance will develop. It just happens more slowly. And that's because those animals that are left untreated, the intestinal parasites that they have are still potentially susceptible to ivermectin, and so the eggs that are being put out into the pasture are still a combination of those that are resistant and those that are susceptible. So it takes a lot longer to get to the point where you're showing resistance in the herd. The last comment I want to make is around environmental impact, and this is the idea. So for those of you who don't know, the average cow produces 65 pounds of manure a day, right? That's a lot. That's a lot of manure going into the environment, and that's on a daily basis. And so if you think about, and the average pig, just for comparison, is 11 pounds. It's still significant. The average human is about a quarter pound. So and we know there's a lot of issues with human contamination of fecal material in the environment, but as far as animals go, cows and pigs are big ones. So when livestock are treated with ivermectin, there's residuals of ivermectin and its metabolites that are put into the environment, right, through manure. And then it goes into other compartments. It can go into the soil as well as into aquatic systems. And we know that depending on environmental factors, which vary across the different areas where malaria is present, the dose or mode of administration, the diet, the animal density, climate, soil type, all of these different things impact the persistence of ivermectin and its metabolites in the environment. And these can have impacts on non-target insects, algae, and fish. And so the indications of these are important for those who are engaged in environmental science and thinking about environmental health. And this is something to consider as we go into communities and do mass drug administration of ivermectin into animals that produce a lot of manure and put it into the environment. So as far as research and development goes, things that need to be considered, we really do need to think about the impact of MDA on resistance to primary helminth targets in livestock, right? This is important to us as veterinarians. It's important to people who have livestock as their primary source of income, and it's important to economies as well as food security. So we need to maintain this as a drug for veterinary use. However, we do need better tools for detecting resistance so we can truly say if it's present or not, rather than relying on the crude tools that we have. We need to ensure that these are aligned with veterinary services and programs. So as we go through and develop, and we're really focused on this outcome of malaria, reducing malaria, and how we are going to dose it, we have to think about the fact that this is being used and dosed, and how might we ensure that there's synergy between animal health uses and the regimen that we think has the greatest impact on human health. And then finally, considering the differential impact across environmental conditions. So in the different areas, in the different countries that still have malaria transmission, and we're thinking about using mass drug administration, how might the environmental conditions impact degradation of ivermectin and its metabolites in the environment, and what secondary impacts could that have on environmental health? So in summary, if we want to use this in livestock, which I think has the potential to be a great idea, especially considering the potential secondary economic benefits, we really need to address all the issues I just discussed today in trials that have clear endpoints, our endpoint of interest, which is malaria, cases or incidents of malaria. We need to look at environmental impact. We need to think about how to finance and deliver this in a way that works for our endpoint of malaria, but is consistent and collaborates with ongoing animal health programs that exist. With that, I'd like to thank the other road mappers, and thank you for your attention.
Thank you. So it was great to have a veterinarian fleshing all of the complexity in these issues out. I do have one question in case anyone else comes up, and that is, under the status quo, not for malaria, how much ivermectin is being used for mass campaigns of herds in the north or the south, any place you have data?
So that's a good question. That would be a good systematic review to complete. I can say it's done intermittently. So it's challenging. If we in the health field, in the human health field, think that human health is underfunded, then you should try working in animal health. It's even more underfunded. So ivermectin is used widely in different ways. I would say in developing countries, it's often more used. You can just go and purchase it at any pharmacy. So people are using it more on an as-needed basis rather than in mass campaigns. Although governments do occasionally go in and deliver mass drug administration campaigns as sort of a form of outreach to improve animal health. But as far as being able to give you a broad scale, that's hard to do. Sure, question.
Yeah, thank you. Thank you very much for that very interesting presentation, looking at the other side of animals and veterinarians. I like your closing remarks on the issues that still need to be addressed, but I was also thinking that I have this question regarding when you look at treating some animals and maybe the resistant worms are left behind. Have you contemplated what it would mean if only males were left behind, or only one single female worm, and then the issues of reinfection? Because it seems like for the numbers to work out right for us, we need to put all those considerations into practice, because we do need male worms to mate with the female worms for any outcome of any eggs and all that. And the effect of the drug, is it the same in male worms as it is to the female worms?
Yeah, so I think, so there's going to be a combination of male and female worms in, so the idea of leaving a population untreated means that the eggs that are shed into the environment are going to develop. And so you don't just shed female eggs or just shed male eggs. So by allowing a population to shed eggs into the environment, they're going to develop without any issue. Does that answer your question? I guess I'm a little confused as to what. There would be no strategy to ensure that there are enough female worms or male worms to develop and mate and reproduce. It would be this idea that by leaving someone treated, that would happen naturally. So there's no way to target that. And that reinfection is going to occur regardless. And in some ways, that's what we want. You don't need animals to be completely parasite-free. They just need to be at low enough burdens that their productivity isn't hindered.
Yeah, that is true. But the point I'm trying to make is, until we understand the effect on the different sexes of the worms, and also understand when we leave some animals with the worms, are we leaving just females or males? And I guess there's no way of looking at that question. The big picture is going to be incomplete because we don't have answers to that. And I think there's something that can be considered by... Yeah,
that would be interesting. And, and it actually probably wouldn't be hard to set up a study to look at that component of it.
Okay. Yeah, thank you.
One thing does bring up something of interest, and that is this idea of refugia also kind of helps with the issue of food safety. Right? So if we want to leave a certain percentage of the population untreated, we could leave those animals untreated that we think are going to slaughter soon, or perhaps those that are currently milking. Right? So there's the idea that you could use refugia to reduce resistance, but also use that as a way to manage the concerns about food safety.
Richard Idro from Uganda. Just a follow-up question, and related to both the application of ivermectin in the humans and in the animals. Let's say we are successful in applying the drug and literally eliminating the mosquito types. And what in the environment, something is going to replace it, whether the other species of mosquitoes or some other type of fly. And what are our projections? What are the imaginations? Potentially, what kind of things may replace them? And what are some of the risks which will be associated with this?
Oh boy, that's a tough question. Would you like to answer that?
No, but I think that's an excellent question that we should visit when the whole panel is up here. And I would invite you to join us first when we first come back. Okay, thank you.
Yep, thank you.
Thank you.